Hello, my name is Lama Aliu, and today I'm going to talk to you guys about my enzyme project. I chose to do my enzyme project on lactate dehydrogenase. So, what is lactate dehydrogenase? Lactate dehydrogenase is considered a cytoplasmic enzyme that's found in nearly all living cells in the body. Um, for short, lactate dehydrogenase is considered LDH. So, LDH is considered a oxidoreductase enzyme that catalyzes the conversion of lactate to pyruvate by converting NADH or NAD to NADH, or vice versa, as if pyruvate to lactate and then NADH to NAD. So, what is the function of lactate dehydrogenase? Lactate dehydrogenase works to maintain homeostasis within the cell when there is absence of oxygen. LDH works actually in a anaerobic state and a aerobic state. So LDH is expressed extensively in the body tissue, such as blood cells, heart muscle, liver, serum, the kidneys. So because it is released during tissue damage, it's considered a marker for common injuries and diseases such as heart failure. So looking at the structure of lactate dehydrogenase. Starting off with the coenzyme for lactate dehydrogenase is nicotinamide adenosine dinucleotide, also known as NAD or NADH. For the substrate would be considered pyruvate or lactate, depending on the path it's taking. If it's pyruvate to lactate, pyruvate would be the substrate, or lactate to pyruvate, the lactate would be the substrate and or the product. The secondary structure of LDH is mainly comprised of 40% alpha helices and about 23% beta sheets. So right here I have the APO enzyme of LDH. Up top here I have the human LDH crystal structure, and down here I have the human LDHB crystal structure. So it's considered a tetramer with two subunits, composed of two subunits, the H subunit and the M subunit. Also, this enzyme is considered a homo or hetero tetramer, mainly because the M subunit could consist of 4 M subunits, 4 H subunits, or a mix of the two. So looking at the structure of the gene, the LDHA gene is located on chromosome 11, short arm P, right over here, and region 1, band number 5, and subband number 1. And down here we have LDHB gene that makes the LDHB or M chain. Oh, I'm sorry, H chain or H subunit. This gene is found in chromosome 12, short arm P, region 1. Band number 2 and subband number 1. So here we're going to look at the secondary structure. First we have the LDHA chain. As you can see here, looking at this, you have about 332 residues on the secondary structure for LDHA. It's considered a polymer with 332 residues. Polypeptide chain. It has around 44% it's 44% helical and 15% 44% helical with 15 helices, which is composed of 149 residues, and about 18% beta sheets with 16 strands, and which is comprised of 62 residues. And this is the LDHB chain. Looking at the secondary structure, here you see the alpha helices, and with the yellow arrows here, you see the beta sheets. So the LDHB chain is comprised of about 334 amino acids, and it's also considered a polypeptide chain. It has a couple more amino acids than LDHB chain, and it has more beta sheets than LDHA chain. So lactate dehydrogenase, like I said earlier, is composed of isoenzymes, specifically 5 different ones. As we said, you have LDHA and LDHB. LDHA gene makes up the M subunit, and the LDHB chain or gene makes the H subunit. So if you look here, you have the first LDH1 isoenzyme. It's composed of four different H subunits, which is mainly found in the heart. And then you have down here you have the LDH5, which is also one of the most common isoenzymes that is found in the muscle, composed of 4 M subunits. And then here you have the intermediate activity determined by the LDH2, 3, and 4, which are found in different areas of the body. Like LDH2 is mainly found in the serum. LDH4 is found in the kidneys, and you have LDH4 is found in the liver, and LDH3 is mainly found in the kidney. And as you see here, LDH1 is mainly known for lactate oxidation, turning lactate into pyruvate by NAD to NADH. And down here you have the LDH5 isoenzyme that is known for the pyruvate reduction, which is pyruvate to lactate. I'm going to answer back to the last slide. Both LDH1 and LDH5 share about 25% of the amino acid sequence. And that's it. So the next slide. The next slide has the catalytic binding pocket here. As far as the catalytic binding pocket for lactate dehydrogenase, it's composed of pyruvate as a substrate, NADH as the coenzyme, and mainly or predominantly using the histidine 195 residue, arginine 109 residue, arginine 171, and aspartic acid 168 residue. These are the residues that are commonly found in the catalytic reaction that we'll be looking at. So the mechanism for lactate dehydrogenase. Here I have the mechanism for pyruvate to lactate, which would be pyruvate reduction. We start off with the NADH. The NADH has the direct hydride transfer from NADH to pyruvate's carbonyl carbon right here. And the carboxyl group then takes the proton that's donated from the histidine 195 residue here, which is facilitated by arginine 171, eventually getting the product lactate or L-lactate. So for the literature, I chose serum LDH and chronic cough, a potential marker of airway inflammation. It was done by Ferrucci et al. 2011, in the Department of Biochemistry in Hull Royal Infirmary in the United Kingdom. This was basically a cohort study, and the objective for the study was to confirm that patients with a chronic cough have elevated levels of LDH. So for the materials, they started off with getting 83 patients and determining what patients they were going to use. So the patients with chronic cough they used patients that were referred to the whole cough clinic that had a chronic cough initially. So for this study, the chronic cough was defined as lasting for more than 8 weeks. And the patients were all new referrals and were sequentially and prospectively recruited. The patients with conditions known to be associated with raised LDH levels were excluded, so they didn't mix up the data for this, and they had more lucid data. So they had some different methods for this experiment. They had a spirometry, which is the measure of how you're breathing, basically. They had the subject perform the 3 forced expiratory maneuvers from total lung capacity to residual volume. And then they had the forced expiratory volume in 1 second and forced vital capacity that were recorded. And the best value for each was used in the analysis. And another method that they used in this study was the LDH isoenzyme assay. This was basically to determine which isoenzymes were found in the, in the patient and in the affected area. In this case, the lungs. So they used electrophoresis, which was performed using the hydrogel 15 iso LDH kit. And after the electrophoretic separation in a alkaline buffer, a series of linked reactions starting from the conversion of lactate to pyruvate by any and all LDH isoenzymes present resulted in this deposition of formazan, which is blue in color, in a gel, allowing the 5 isoenzyme fractions to be quantified by densitometry, which is determined by the isoenzyme's electrophoretic mobility. So a couple other methods that they provided here or in the study were one, the cough symptom scores and the cough questionnaire, and then they kind of saw the correlation among the 2 scores. The cough symptom scores was just having the subjects, or asking the subjects the severity and frequency of the coughs and scoring them from 0 to 9. And then for the cough questionnaire, the subjects were asked to complete the whole airway reflux questionnaire, which is a 14-point self-administered questionnaire with a maximum score of 70. And this was just basically a small determination of your kind of how severe your cough was and how frequently the cough was for the patient. Some of the results they found, starting off with the scores, with the questionnaire and the score, they found a correlation, and they found that there was a correlation between the severity and the questionnaire scores that they had. They found a p-value of less than 0.001, which you could say that you could reject the null, and there's a significant value there. Here we have a graph showing the different isoenzymes, and on the x-axis here, and on the y-axis you have the LDH in units per liter. They found that there was an increase in the isoenzymes LDH 3 through 5, and almost or more than half of the patients that they recorded after an 8-week period. Here in the bar graph, you'll see the dark bars indicating the laboratory reference. That's basically when they started. Oh, I'm sorry, no, the reference is the what they were kind of maximum, what they were expecting from the patient as far as the LDH in units per liter in this serum. As you see here, the predominant ones were LDH 3, 4, and 5. The chronic cough bar here, which is indicated by the silver or light gray color here, kind of are a lot higher, a lot higher than the reference number they were expecting. Here we have another graph here. So the mean LDH values that were recorded were 474 starting at the baseline at the beginning of the 8 weeks. It was 474 units per liter. And then after the 8 weeks, the mean LDH was 486 units per liter, which gave them a p-value of less than 0.01, which indicates a significant value there as well. Looking at the graph here, though, you'll see that the x-axis is the baseline and the y-axis is the LDH levels at 8 weeks. And one of the big ones here, you'll see that around 300 units per liter at the baseline, you'll see most of the patients starting around 350 and ending after 8 weeks around 600 units per liter. And then there's a couple other examples in here that also back the data up there. Here's the third bar graph that I included. This was the laboratory reference range that I was talking about. The laboratory reference range was 215 to 485 units per liter that they were expecting, or kind of referencing as far as range goes. And more than 3/4 of the patients had a serum LDH value in the 4th quartile of the reference range or above. As you can see here, the x-axis is measuring the quartiles, and the y-axis is measuring the percent of LDH in the serum. And then, obviously, the distribution of the interquartile reference range. As you can see, as I said, in the 4th quartile, you see a significant value there. It's pretty high up there, which more than half the patients fell into the 4th quartile or above. So the conclusion for the paper was that serum LDH levels are elevated in a substantial proportion of the patients with chronic cough. This rise is likely to be due to airway inflammation, known to be associated with chronic cough, also due to, which was measured by the LDH levels. Thank you for listening, and these are my references right here. Thank you. There's something I wanted to add, but I didn't.
Is there like
anything
is too long? It is long.
Really?
It was like 20 minutes.
Ah, shit.
Is it too long?
Yeah.
So we gotta do it again?